As mentioned, I'll be talking about the magnesium carbonate mineral formation potential of sulfate-reducing bacteria, and this work was supervised by Janine McCutcheon and was through the University of Waterloo. But first, I would like to begin by acknowledging the land on which I am currently residing, as well as the land on which I conducted this research. The University of Waterloo is located on the traditional territory of the Adwandaran, Anishinaabe, and Haudenosaunee peoples. Our main campus is situated on the Halaman Tract, the land granted to the Six Nations that includes 6 miles on each side of the Grand River. The research was conducted on the traditional territory of the Huron-Wendat, Anishinaabe, and Mississauga peoples. So industrial activities generate metal-contaminated sites and acidic waste, creating the need for remediation efforts. Microorganisms such as sulfate-reducing bacteria, or SRBs, have proven useful in bioremediation of industry waste, partially due to their role in mineral precipitation reactions. They produce negatively charged extracellular polymeric substances, or EPS for short, which help them to grow and survive in ion-rich environments. The functional groups of these EPS also help to bind positively charged ions from the solution, which then contributes to mineral precipitation. We know that SRBs have played a role in the production of carbonate minerals previously, primarily calcium carbonate minerals, but we also know that SRBs are well known to precipitate sulfide minerals by the reduction of sulfate. In that process and during their metabolism, they also generate alkalinity, which can result in a pH increase of the system, and this can directly contribute to the precipitation of carbonate minerals by inducing supersaturation conditions. So for example, we can see that SRBs are largely responsible for the cementation of organic matter produced by cyanobacteria in the process of stromatolite formation. And while these studies do a good job of demonstrating the role SRBs play in calcium carbonate mineral precipitation, their role in precipitating magnesium carbonate minerals is not really well understood. So that was kind of the goal of my research, was to first of all determine the ability of SRBs to precipitate magnesium carbonate minerals, especially in highly concentrated solutions that would be representative of what could be leached from mine tailings. And the second goal was to identify any types of biogeochemical limitations on this mineral precipitation process. Hopefully, the results of this experiment would lead to a better understanding the role of SRBs played in carbonate mineral precipitation in both calcium and magnesium-rich environments, such as mafic and ultramafic tailing storage facilities. So the study area selected for this site was a circumneutral tailings pond situated next to a limestone-treated tailings pile in Sulphide, Ontario. This is a very small area, so if you don't know where that is, that's northeast of the township of Tweed, Ontario. The tailings pile was a conglomeration of tailings taken from historic pyrite mine sites in that area. Historical records indicate that mining took place in that area between 1877 and 1916. However, the actual composition of the tailings was not really well historically documented. So in image A, we can see the tailings pile and the tailings pond located directly downslope of that tailings pile. And that's where I did primarily most of my sampling. So along the shoreline, I found SRB-rich sediments, which we can see in image B. Those darker sediments have a high concentration of sulfate-reducing bacteria in them. And these were collected and stored in 50 milliliter Falcon tubes and remained anoxic until returning to the laboratory. Once at the lab, these were cultured using modified postgate media, and SRB enrichment cultures were created. So from there, 28 160 milliliter vials were created replicating synthetic mine wastewater. So 2 different solutions were created. The first one contained higher concentrations of magnesium, which I'll refer to as solution A, and the second contained higher concentrations of both magnesium and calcium, which I'll refer to as solution B. Each solution also had an abiotic control associated with it. So for the magnesium-rich solution, that will be referred to from here on out as solution AA, and for the high concentration of magnesium and calcium solution, I'll refer to that as solution BA. So these solutions were inoculated, And after about 2 weeks of growth, we could see clear visual differences in both solution A and B from their abiotic controls. So as seen on the slide, there's a clear cloudiness shown in both of the biotic solutions, while the abiotic solutions remained clear. We also monitored any biomass production or mineral precipitation that occurred using scanning electron microscopy, and we took a micrograph of the initial SRB and their consortium, and we can see some mineral precipitation already occurring. We also monitored the fluid chemistry by measuring pH, conductivity, dissolved oxygen, alkalinity, the major cations, major anions, including the organic acids, and dissolved organic and inorganic carbon content. We also measured the chemical composition of mineral precipitates using EDS. The way that we sampled for this experiment, as it was conducted over 11 weeks, was measuring a subset of vials each week, and we used sacrificial sampling, whereas one vial per week would be essentially sacrificed for measurements in order to keep the remainder of the vials anoxic for the entirety of the experiment. So when comparing our water chemistry data, we plotted the abiotic control, the similar results this way. So as we can see, all solutions began with a very high concentration of lactate when talking about. However, over the first 2 weeks of the experiment, which, as mentioned previously, this was the growth phase that I had for my bacteria, almost entirely all of the lactate was consumed down to 0 milligrams per liter. To me, this indicates that the lactate was being consumed as an electron donor for the sulfate-reducing bacteria, and then during their metabolic process, they produced formate and acetate. That in both biotic solutions, the lactate remains within the solution and is measurable, and we don't see the same production of formate and acetate. And there wasn't really any other notable changes in the rest of the organic acids for this experiment. When considering pH, DIC, DOC, and alkalinity, we can see that the DIC concentrations of both of the biotic solutions was much higher than that of the abiotic counterparts, and the opposite was true for the DOC concentrations. The DIC trend indicates in both cases in solution A and B that there was likely carbonate mineral formation over the first 5 weeks, as we can see that it's increasing. And then after the first 5 weeks, this kind of decreased and was a lot less likely to be formed. We can also see that the alkalinity in solution A and B was much higher than those of the abiotic counterparts and also kind of fluctuated on a weekly basis. For the abiotic solutions, both of the alkalinities remained the same and constant over the whole 11 weeks of the experiment, indicating that there wasn't any real microbial growth that would be influencing the alkalinity. From the alkalinity's fluctuations, as well as the high alkalinity concentration, we can determine that this was likely biologically induced alkalinity. And it's also important to note that the pH throughout the experiment remained between 5 and about 6.25, which is outside of the typical range for carbonate mineral precipitation. When comparing our anions and cations, this is only speaking towards the inoculated solutions. So we can see that in terms of magnesium in solution A, we generally see a decrease in magnesium over the course of the experiment. Whereas in the solution B, which had both a high concentration of magnesium and a high concentration of calcium, we see that there is a decrease over the first 5 weeks of the experiment, and then it begins to increase again. And we can also see a similar trend in iron, sulfate, and sodium within both of these vials. So we can note that sodium and iron were likely sorbed from solution onto cells or EPS as the biomass increased over the first 5 weeks. and then may have desorbed back into the solution as the active cells or the EPS degraded. And then we can also note that in terms of sulfate, over the first 5 weeks of the experiment, it's indicative of the SRB's metabolism, which would be reducing the sulfate and converting it into hydrogen sulfide. So when measuring the precipitates that form during the reactions, we can see that in the first 5 weeks of the experiment in both solutions A and B, we can see this flaky precipitate that is formed, and those are corresponding to images A and B on this slide. These flaky precipitates in solution A over the first 5 weeks contained very high concentrations of iron and silicon, and these were similar to those found in the initial SRB enrichment culture. In later weeks, which is shown in image C, precipitates generally had a higher concentration of magnesium, sodium, and iron. And it's also important to notice when comparing image A and C and image B and D that these flaky precipitates are not as common in the later weeks, so week 11, which is the endpoint of the experiment, likely indicating that they had either dissolved back into solution or other precipitates were much more commonly formed. And in week B, we can see that generally the flaky mineral precipitates are high in iron, and then in later weeks, the precipitates that have formed are higher in iron, calcium, sulfur, and sodium. It's also important to notice when comparing images A to C and B to D that there are much less active or intact SRB cells than there are in later weeks. So in images A and B, we can see the Vibrio structures of SRBs remaining relatively healthy. And if you're not familiar what Vibrio cells look like, they're kind of like the worm-like structures that we can see throughout the SEM image. And in later weeks, we see much less of those, or they appear almost kind of desiccated, or they're much skinnier, less kind of plump as they appear in earlier weeks, indicating that they probably just aren't as active or aren't as healthy. And we see more coccoid cells appearing, indicating that this might be another type of bacteria or a heterotroph that has taken over in the metabolism. So some of the key findings for this experiment, based on the fluid chemistry as well as the relative abundance of SRBs that we see, that the SRBs only remain the dominant metabolism for approximately the first 5 weeks of the experiment. And then after these first 5 weeks, it's likely that another heterotroph took over, and it was likely present from the initial consortium from the mine site, but then was able to dominate in the solution. So we also noticed that minimal magnesium was measured in the observed precipitates, although we started with very high concentrations of magnesium. So this indicates there is some type of limitation of SRBs to form magnesium carbonate minerals in these situations. We also noticed that in solution B, we began with very high concentrations of magnesium and calcium carbonate, and calcium, which indicates that there might also be a limitation in forming calcium carbonate minerals as well. So this could be due to nutritional limitations. As mentioned before, lactate was entirely consumed within the first 2 weeks of the experiment, and we did see less intact SRB cells after the first 5 weeks. So this might indicate some cells have died off. We know that the DIC and alkalinity have increased in the first 5 weeks of the experiment, which is consistent with SRB activity and should have contributed to carbonate mineral precipitation. However, this isn't necessarily what we saw when looking at the precipitates. And this might also be due to the fact that the pH never increased above 6 in the biotic vials. The closed system of the vials was in order to keep everything anoxic, but however, this might have also contributed to the inability to degas hydrogen sulfide, which would lead to the loss of protons and might have increased the pH if this proton could be lost, and would then perhaps allow the solution to be within the suitable range for carbonate mineral precipitation. The lack of precipitation also indicates that the SRBs may just rely on a relationship with other microbes like cyanobacteria in order to form carbonate minerals. But overall, these findings indicate that there are some type of nutritional and environmental constraints on the carbonate mineral precipitation, specifically magnesium carbonate mineral precipitation, and this will ultimately influence the ability of SRBs to be used in the remediation of industry waste. So I have some next steps for this experiment in order to try and kind of figure out what some of those limitations might be. So the first step is to do XRD analysis of the final week of mineralization. So currently I'm in the process of doing this. However, very little precipitate was actually formed during the experiment, as well as it's important to keep the precipitate that did form anoxic so that no new minerals precipitate in the process of analyzing these precipitates. So that's kind of a problem that I'm currently working around in order to find out what exact minerals are in the precipitates. I'm also planning on doing bacterial community composition analysis in order to find out what strain of SRBs are within the sample, as well as finding out what those other heterotrophs that existed within the consortium could be. This might also contribute to the understanding of the nutritional limitations, as we know from previous work that certain SRBs prefer certain electron donors, and though depending on which electron donors are used, this can also influence the pH, where certain electron donors will allow for an increase in pH and certain electron donors will cause a decrease in pH. So that's also something to consider. And then I also plan on focusing on the mat layer relationships to mineralization and the water chemistry over depth and time. So focusing on a mat layer similar to the image that you see on this slide, where the water chemistry will be measured at each layer of the mat, as well as the mineralization will be considered at each layer of the mat, and this will run for several weeks in order to tell if we can characterize new precipitation that's occurring. So I just want to thank you for inviting me here, and I would like to just acknowledge the University of Waterloo, as well as my supervisor and my committee with their help with this project.